I'd like to start off by thanking the conference organizers, particularly Dr. Furman, for the invitation to join us here today. I'll be speaking about next-generation sequencing in CLL. I have no disclosures. In 2018, despite our increasing reliance on next-generation sequencing technologies in clinical cancer practice, all too often the typical NGS workflow looks something like this. We get a tumor sample from a patient, and we send it off somewhere where it gets put into a black box where something mysterious happens, and then, as if by magic, a clinical report is generated. And while I say this half in jest, considering that differences in next-generation sequencing methodology can have important consequences for downstream interpretation in clinical practice, for the next few minutes, my task will be to try to unpack this genomics black box and to make its contents a bit more accessible. We'll start off with the observation that all next-generation sequencing follows the same general workflow. You start off with genomic DNA, which is then isolated into a library of DNA fragments. And by the term library here, I just mean that adapters are added to the ends of the fragments to make them compatible with a sequencing machine. This sequencing machine then converts the nucleotide biological information into digital information in the form of a large text file that consists of individual reads. And those reads are then processed through various computational analyses to derive genomic variants. And it's this workflow that's going to form the outline of my talk, although I'll start at the beginning briefly with just a primer on the CLL genomic landscape. Then we'll do a very deep dive into the next-generation sequencing workflow, and finally, we'll conclude by looking at some examples of NGS that are as in current clinical use and also some potential uses for the future. We'll start with the CLL genomic landscape. Next-generation sequencing has revealed that CLL is a very heterogeneous disease from a genetic perspective, and that CLL lacks a single disease-defining mutation. Large studies of international patient cohorts have shown that few mutations occur across patients at a frequency greater than 5%. And these studies have also shown that there's a long tail of driver mutations, all with a frequency under 20%. They've demonstrated that the background tumor mutational burden in CLL is relatively low compared to other malignancies, with a tumor mutational burden of about 0.6 mutations per megabase. They've also identified top CLL drivers, including TP53, Notch1, SF3B1, ATM, and BRC3. And they've also shown that approximately 60 to 70% of patients carry at least one driver. The genomic aberrations that show up in CLL affect a select number of cellular programs, including Notch signaling, DNA damage repair, and RNA processing. And for an excellent review on this subject and the particular importance of select genetic aberrations, I point you to a review in the ASH Education book this year published by Drs. Giordano and Dr. Rossi, who spoke earlier. With that, we'll move to a brief introduction to next-generation sequencing in CLL. So a good place to start is to ask, what's so next-generation about it? Certainly, all DNA sequencing follows the same general pattern of fragmentation and subsequent amplification before sequencing. And the key difference to hone in on is really the throughput. Whereas first-generation sequencing, or Sanger sequencing, was serial in nature and low throughput by definition, next-generation sequencing is massively parallel and high throughput. And I'll talk more about this in detail. What you're looking at here is the output from a sequencer. This is a raw sequence file with each row representing a read of an individual DNA molecule on the sequencer. This text file can be huge. It can be millions of lines deep. And it's this raw data that lies at the foundation of all clinical genomics. The computational analysis extracts this clinically useful information, and it does it from the ground up. These raw reads are first aligned to a reference genome. Variation from the reference genome or a germline sample ends up dictating true variants, and these variants are annotated according to their clinical significance. This brings me to the first key concept that's crucial to understanding any NGS assay, and that's the concept of coverage. Coverage, or depth, is defined as the number of unique reads that include a given nucleotide in the aligned sequence. And so here at the top, you can see we have our reference genome. We'll align some reads to that reference genome. And then we can ask, at each particular locus, how many aligned reads are at that location. And you can see in this example, we have 0 reads over here and 5 reads over here for a coverage of 0 or 5. And coverage is important because as it gets deeper, it improves the sensitivity of calling variants and enhances detection of tumor heterogeneity. And we can use a similar sort of approach to call cancer genome variation and infer it from these sequencing read alignments. For example, here, we could detect single nucleotide variation. So you'll see in the reference sequence we have up at the top, there's an A in the germline reference. And you can see that our aligned reads here have a C. Thus, we've detected a point mutation. We can use a similar strategy to pick up on small insertions or deletions. Or we can look at larger genomic intervals and compare the number and proportion of reads that align to the reference to pick up on copy number alterations. Specifically, we can look at deletions, which may be homozygous or hemizygous, or we can look at gains. Gains can be small, such as focal amplifications in genes, or they can be large, up to the size of entire chromosomes, such as trisomy 12. Finally, we can look at structural variation. So when we have one read where one end aligns to one chromosome and the other end aligns to another chromosome, as shown here, chromosomes 1 and 5, we've detected structural variation in the form of a translocation breakpoint. Now we'll dive deeper into the next generation sequencing workflow. Of course, this starts with sample acquisition. In the case of CLL, that's a blood draw, which is usually subject to further processing in the form of a fight call. Peripheral blood mononuclear cells are then subjected to additional purification and isolation of tumor through a CD19 selection. DNA is then extracted from the tumor cells, and then that DNA is fragmented and made into a library. Now here we have a choice. We can either load the DNA from the tumor cells in an unbiased fashion onto the sequencer, or we can do an enrichment and select for particular subsets of information, and this is what's done in the case of whole exome sequencing or targeted sequencing. Regardless of whether we do an enrichment, we load that DNA library onto the sequencer, and this is where the magic really happens, where that nucleotide information is translated from biological information into digital information, and that large text file of reads is output. The individual reads are then aligned to the reference genome, and various computational packages called variants. These variants are then reported and subject to downstream either automated or manual interpretation, and the clinical report is generated. And so this process really takes place in 4 phases. The first phase is in the pathology wet lab. The second phase is on the sequencer. The third phase is the bioinformatics pipeline, and I'll talk about that in more detail. And the fourth phase is interpretation and clinical reporting. Now I'll just pause briefly to talk about target enrichment. Because human beings are notoriously bad at thinking in terms of orders of magnitude, I've created this map visualization to let us compare the relative size of different genomic assays. So for example, if we start off with the whole human genome, the haploid genome, that's exons plus introns plus intergenic regions, about 3 gigabases, 3 billion bases worth of information, and we set that equal to the surface area of the Earth, we can then ask the following question: how big is the whole exome, or just the coding portions of DNA? The whole exome is about 50 megabases in size, and on this scale, that ends up being about the area of the continental United States. Furthermore, we can compare a whole exome sequencing to targeted sequencing, in which we enrich for selected exons or hotspots. And so how big is a typical targeted panel? Well, the one we use at Cornell is around 250 kilobases, which on this scale is the size of Maryland. And it's this significantly reduced region of interest that allows us to have deeper coverage. And the reason we can do that is because there is a fixed amount of sequencing capacity on the next generation sequencer. So for example, we could spend that capacity in different ways. We could either perform whole exome sequencing, look at 50 megabases worth of information at 100x coverage for a total amount of covered material of 5 gigabases, or we could focus on a much smaller region of interest, 250 kilobases, cover that information at 15,000x coverage, and we have the same amount of real estate on the sequencing machine. In general, these assays can be categorized as either broad or targeted. And in the case of broad assays, they are traditionally research-focused. They're well suited to discovery applications, and in the case of whole genome sequencing, they're unbiased, but they both provide extensive information. And in the case of both whole genome and whole exome sequencing, this greater breadth of information is sampled at lower coverage, which can limit detection sensitivity. Whereas targeted panels are primarily used in the clinic. They're ideal for well-defined use cases where you know exactly what it is you're looking for, and they typically have higher coverage of the regions of interest. And deeper sequencing in targeted panels improves the sensitivity of calling rare variants. So now we'll look under the hood of the next generation sequencer. So this is really the amazingness of next generation sequencing. It's what happens on the sequencing machine, and I'll try to explain it right now. So as I said, the first step is to fragment the DNA into fragments that are compatible with the sequencing machine. So there are adapters added to the fragments. Those adapters allow the fragments to attach to a glass slide called a flow cell, where the sequencing reaction takes place. The first step is amplification of that individual fragment, and then sequentially, the next generation sequencing machine injects a DNA polymerase along with color-labeled nucleotides. As a nucleotide is incorporated into the growing DNA strand, it emits a color of light that's captured by a camera, and the particular nucleotide identity can be inferred by looking at the colors of the pixels in that image. We can then infer the entire nucleotide sequence by looking at the changes in colors across a series of images taken by the camera. That brings me to the computational pipeline. Again, I remind you that the raw reads form the basis for the input to the computational pipeline, which are then aligned to a reference genome. And that information is subsequently used to extract all of the copy number variations, single nucleotide variants that we want to produce in a report. The pipeline is just a series of computational packages that tie together to process this data. So for example, there are different computational packages that do the alignment and different computational packages that do variant calling. Another way of looking at this is that the information comes off the sequencing machine in the form of a text file. It's aligned and goes into another file format, which can then be passed to individual computer programs that do the variant calling, resulting in a VCF file which contains information about genomic variation. Before I talk to you about exactly how to interpret the variants, we should look at one use case of variant calling in particular, and for that I'll focus on single nucleotides. Here I want to distinguish between SNPs and SNVs. So when I talk about single nucleotide polymorphisms, we're really talking about point mutations in populations. These are the point mutations that constitute 90% of all genetic variation between humans. When we talk about single nucleotide variants, we're talking about point mutations in somatic cells, including tumors, and these are the things that we're interested in. There could be different classes of point mutations in coding regions. For example, a mutation can be missense, in which a single base substitution alters the amino acid sequence of a protein. They can be silent, in which that substitution doesn't change the amino acid sequence. Or they can be nonsense or truncating, in which the single base substitution introduces a premature stop codon. Here's an example of the results of an output of a variant caller. And you can see in this case, we've identified somatic variants in 3 genes, BTK, TD53, and Notch1. And you can see that each of these mutations is listed along with its class. So for example, starting with BTK, you can see that we have a mutation of a single nucleotide residue. And that nucleotide residue affects the amino acid at position 481 in the protein. In this particular case, a cysteine residue is changed into a serine residue, and that's a missense mutation. And what also shows up on the output of a variant caller is a percentage called the VAF, or variant allele frequency. And this is a commonly misunderstood measure, and I'm going to go into more detail about it right now. A VAF is defined as the variant mutant reads over the total number of reads. So in this example, I will discuss a case of 5 total cells, 4 of which are tumor, seen here, 1 of which is non-tumor, for a total sample purity of 80%. And in this example, you can see that for 2 genes, each of 2 genes, there are 2 alleles, 1 maternal and 1 paternal. And if we start by looking at SF3B1, you can see that each of the tumor cells is a heterozygous mutant, having 1 mutant allele. And thus, to calculate the VAF, we take the total number of variants, that's 4, 1 for each cell, and we divide by the total number of reads, that's all the alleles, or 10, and we can see that we have a VAF of 40% for SF3B1 mutation, which reflects a heterozygous mutation in 80% of all the cells. Now if we switch and look at Notch, you can see that I've altered the example such that this tumor cell here on the bottom is actually wild type for both Notch alleles. And by doing that, essentially I've introduced some tumor heterogeneity. And so now when we calculate the VAF, we calculate the number of variant mutant reads. That's only 3 in this case for the 3 mutant alleles in these tumor cells, divided by the total number of reads, again, that's 10. And so we get a VAF of 30, this time reflecting a heterozygous mutation in 60% of total cells. And so the key takeaway here is that the VAF as a measure is not only dependent upon the allelic burden of the cancer, but it's dependent upon the tumor purity and tumor heterogeneity. And we can think about this from a statistical sampling perspective. It's like pulling marbles out of a bag. If you want to detect a lower allelic frequency, you need to pull more marbles out of the bag. Deeper sequencing is equivalent to more sampling, which is equivalent to a better ability to converge on the underlying truth. Another way we can look at this is to compare the confidence of detection at different depths. So in this example, we could compare whole exome sequencing versus a deep analysis of circulating tumor DNA. Just to orient you, on the x-axis here you have depth, and on the y-axis you have the sensitivity and the ability to call particular variant allele fractions. So you can see that whole exome sequencing typically proceeds at a depth of about 100x, and there we can feel confident calling variant allele frequencies of 10% and above, whereas a circulating tumor DNA type assay, we might sequence very deeply over a very small area at a 25,000x and feel confident calling variant allele frequencies of 1% and above. Another way to look at this is to look at sensitivity versus breadth and ask which assay is right for your application. The answer to that question really depends on what it is you're interested in. If you're interested only in maximizing sensitivity, then NGS might not be the way to go. Techniques like digital and allele-specific PCR methods are the most highly sensitive, but they can detect only point mutations and short indels on a very few number of loci, whereas next-generation sequencing is more versatile and enables querying multiple loci up to the whole genome, detecting point mutations, indels, and copy number alterations, but at a lower relative sensitivity compared to digital droplet PCR, although the sensitivity of NGS is pretty good. And finally, we can take a look at a clinical report. So this is just the output of variant callers addended with a note. You can read the note here. It says that the C481S mutation in the BTK gene has been shown to be associated with acquired resistance to BTK inhibition by ibrutinib. And so we can ask how to interpret these results. And interpretation of results really ends up residing, the responsibility for that ends up residing with the clinician and the clinical expertise and the clinician's due diligence in reading the medical literature. And so finally, I'll conclude with a few examples of applications of NGS and CLL genomics, mostly in the research setting. In this example, which was published in the JCO by Dr. Bird's group, whom we heard from earlier, they implemented a prospective surveillance strategy looking for BTK resistance mutations emerging in patients on ibrutinib. And in this study, they used an assay that covered the BTK loci at a depth of 3,000x, and they had the ability to call a BTK variant VAF of 1% and above. They looked at 122 patients, and over the course of the study, 16 of those patients developed BTK resistance mutations with increasing VAF, and of those, about half experienced clinical relapse. This was done in the research setting, but it does raise the interesting question of whether such type of surveillance could be implemented in the clinic, or at least point out specific patients who should be paid further attention to. Another example of the importance of subclonal TP53 mutations. So we all know that clonal TP53 mutations are associated with an inferior survival. In this study, David Rossi and colleagues, and then subsequently others, looked at distinguishing a clonal TP53 VAF versus a subclonal VAF, and the cutoff here was 12%. Why was it 12%? 12% is the detection threshold for traditional Sanger sequencing. And what you can see in these Kaplan-Meier curves, there is indeed no difference between the adverse prognostic impact of a clonal or TP53 subclonal mutation. And finally, my last example, we can look in the ibrutinib era of an application of next-generation sequencing to detect clonal evolution in patients on ibrutinib. So Dr. Landau and colleagues reported in Nature Communications that they were able to detect clonal shifts early in the course of ibrutinib therapy, and that this was a significant predictor for risk of progression with a hazard ratio of 3. And so I think there can be no doubt that next-generation sequencing will continue to improve and its costs will continue to decrease. And as NGS techniques continue to contribute clinically meaningful information in CLL, I think it's important that we always remember what's under the hood. And with that, I thank you for your attention, and I thank the conference organizers and my Cornell colleagues. Thank you very much.